So my talk today is going to be about how do you know where you are, and how does the hippocampus and its input, together with its inputs, create a representation of space, and how this representation of space gets richer with increasing task demands. So what we're going to do in
Excuse me,
this talk.
sir. Sorry to
Sure.
interrupt. There
Sure.
was a slight mistake. Audience, this is in the last session. There'll be another one right after this. Thank you.
Yeah, thank you. So the talk today, I'm going to talk about where are you now and how do you know that, and the main focus is going to be about how hippocampus creates a representation of space, how it's put together with the different inputs that the hippocampus gets, and how this representation gets more and more nuanced or complex as the task demands increase. So we'll start with a very simple paradigm. We'll see how hippocampus represents space in a very simple task. Then we'll add more task demands, and then we'll see, first of all, what the inputs to the hippocampus do. And then we will see how the hippocampus uses this information to enrich its own representation of space. And then if time permits, we'll also talk about more recent lab research in more complex environments. But let's see how far that goes. This is kind of ambitious to put in too much. So let me just start with telling you what a hippocampus is. Hippocampus is this structure that lies at the core of the brain in the medial temporal lobe. When extracted, the hippocampus looks like this. As you can see, the hippocampus is very similar in shape to the seahorse. And the seahorse itself is named after the mythical horse-fish hybrid, the hippocamp. And that's where the name hippocampus for the structure hippocampus comes from. Now, this little story that I just told you about the name of the hippocampus is in itself a very good parable for hippocampal function. I told you a little anecdote. I told you exactly once this story, and hopefully if you found the story interesting, you're going to remember it for quite a long time in your life. This kind of memory, or the episodic memory, is a function of hippocampus and its inputs, the entorhinal cortex and so on. And it's like, it's one-shot learning. You remember it for a long time, and you require hippocampus for it. So we are not going to talk much about the episodic memory throughout the talk, but you'll see as I'm talking about space itself, the components required for having this kind of memory already start falling into place. So towards the end, if people have questions about how does it relate to episodic memory, I'll be happy to answer those questions, but I'm not going to focus on that today. Today, the focus is about space in the hippocampus. Obviously, I showed you human hippocampus here. It's not possible to do all kinds of complex studies we need to do, invasive studies we need to do with electrodes sticking in the brain and so on human hippocampus. So we use the rodent hippocampus as the model system. Here is a image of a sketch of a rat brain with rat hippocampus, which is now this banana-shaped structure in there. And when you take a cross section through the hippocampus, it looks like this. There's a very well-defined cell body layer, area CA1, CA2, CA3, and the dentate gyrus. So these are parts of the hippocampus. We'll look at some parts of the hippocampus as we talk about recordings from the hippocampus. The hippocampus receives majority of its cortical inputs via the lateral and the medial entorhinal cortex. Pretty much whole lot of brain, most of the brain projects to the hippocampus, but it doesn't project directly to the hippocampus. Cortical areas project to lateral entorhinal cortex and medial entorhinal cortex, and then the information is channeled from these two cortical areas into the hippocampus. And here is a cross section of medial and the lateral entorhinal cortex, showing you different cell layers. This is not your classic neocortex. Layer 4 is the quiet zone. Inputs come in layer 2, 3, 2, 3 projects to hippocampus, and feedback goes to layers 5, 6, and so on. So when we look at the network that involves hippocampus, here is what it looks like. This is just to give you an idea about the parts of the circuit. Don't worry about specific names. Don't worry about, oh, here are small functions associated with each individual area and so on. We'll talk about specific regions and specific inputs, what they do and so on as we come to that. So don't worry about the names. Much of the information that comes to hippocampus, as I said, gets channeled via lateral and medial entorhinal cortex, but the information seems to be somewhat segregated between lateral and medial entorhinal cortex. Just like you heard in previous talk about what and where, there seems to be segregation about sensory information versus path integration or movement derived spatial information between lateral and medial entorhinal cortex. When this information comes in these two regions, it's segregated between the two. But when it comes to the hippocampus, between dentate gyrus and area CA3, both of them project to the same parts of dentate gyrus and CA3. So the information merges together over here. There's a lot of recurrent connectivity both within dentate gyrus via hilar neurons and CA3 with CA3 pyramidal cells projecting onto themselves. And then the information gets to area CA1 of the hippocampus, but it's kind of complicated there, wherein it's merged information coming in via CA3 that projects to both CA1 proximal and distal, and segregated information projects from lateral entorhinal cortex to CA1 distal and medial entorhinal cortex to CA1 proximal. And we'll see in the second half of the talk how this affects the information that is represented in these parts of area CA1. But to begin with, we will talk about how the hippocampus represents space in the activity of its neurons, the pyramidal cells. So the experimental paradigm for this is simple. We have a drive implanted which projects a bunch of tetrodes in the rat brain. These tetrodes are targeted at different parts of the hippocampus. So in this experiment, area CA1 of the hippocampus, where the pyramidal cell layer is marked over here. So you project an electrode into the pyramidal cell layer. Hopefully your screen resolutions are high enough for you to see a tetrode projecting out here. So as you can see in this drive, one screw has been turned down such that one tetrode projects down, but this drive has 16 tetrodes and a couple of references to target multiple neurons in the hippocampus. I mentioned the word tetrode, so I might as well quickly tell you what a tetrode is. Tetrode is bundle of 4 individually insulated wires. So there are conducting cores, each of which is insulated with an insulator, and they're twisted together. These are of the order of 12.5 micron or so electrodes. And so they are of the order of size of a cell body. When you project these electrodes in the CA1 cell body layer, you are able to record from large number of neurons simultaneously and at the same time tell apart their activity from one another. For example, neuron A that is closest to wire 1 will give rise to an extracellular signal every time it fires an action potential that is largest at wire 1 and smallest at wire 4. In contrast, neuron D, which is closest to wire 4, will give rise to an action potential extracellular signal that is largest at wire 4 and smallest at wire 1. And therefore, when you plot peak height on wire 1 versus peak height on wire 4, the action potentials of neuron D will clearly segregate from action potentials of neuron A. And you can use a whole bunch of these parameters to segregate activities of large number of neurons that are recorded simultaneously. Given the cell density of hippocampal cell layer, you can segregate activities of anywhere between 5 and 30, 40, 50 neurons on a single tetrode recorded simultaneously. And because there are multiple tetrodes, you can record from dozens to hundreds of neurons simultaneously as the rat runs around performing all kinds of tasks. So you can ask, what are the ensembles of neurons doing as the rat runs around? So here we'll see a video. Um, yeah, we'll see a video of a rat running around. We have electrodes stuck in the hippocampus. You're going to hear some background activity, and you will hear sharp clicks every time the neuron fires action potentials. You've heard these kinds of clicks in the earlier talk, so hopefully this won't be too much of an effort to figure out what is going on. Just keep an eye on where the rat is and what it is doing as you hear these action potentials. So here comes the rat, it turns around, it gets into the place field of this neuron, and you hear a lot of clicks. Yeah, there you go. So every time the rat is at this location approximately, the neuron fires a lot of action potentials, but when it's elsewhere, it is either very quiet or fires a very few action potentials. So now you can create a rate map of this activity in order to figure out where does this neuron fire, where does this place cell, as it's called, fire. Okay, and as you heard, a lot of action potentials happened here. So if you divide the number of action potentials at each location by the amount of time the rat spends at each location, you will get a firing rate map. Here you'll see firing rate is highest over here and lower over here. And you can ascribe pseudo colors to indicate firing rates. So here, for example, here is a example neuron with a place field in the middle of the square environment, such that the neuron fires a lot in the center, where the activity is represented by this dark red. And as it goes away from the center, as the rat goes away from the center, the activity of the neuron drops such that the firing rate gets lower and lower, and that is represented by cooler and cooler colors. So you go through yellow, cyan to blue, and the deepest blue indicates locations where the neuron does not fire any action potentials at all. So now you can look at what do multiple neurons recorded simultaneously do. I showed you activity of one single neuron and showed you how that particular place cell fires at one particular spatial location. But there are thousands, tens of thousands, or hundreds of thousands of neurons in a given region of hippocampus. What do they all do? So here is simultaneous recording of 80 neurons. This was one of the first, it was rather the first recording of large number of neurons in hippocampus done by Wilson and McNaughton in '93. So each of these squares represents activity of one single neuron in a square and in a particular square environment. So different squares are activities of different neurons recorded simultaneously in exactly the same environment. So you can see this particular neuron fires towards bottom left of the environment. This neuron fires to the center right of the same environment. This neuron fires at the exact center of the environment. There are neurons that fire at top right corner of the environment, middle left of the environment, and so on. So all the neurons recorded simultaneously cover the entire environment, such that just by looking at the activities of the neurons, you will be able to pinpoint the exact location of the animal at any given moment. This is pretty much akin to a GPS telling you exactly where you are on this map of Bangalore. So right now, for example, I'm giving this talk from my home, which is in Malleshwaram, and IISc is somewhere around here, my workplace, and my neurons in my brain will be representing this location. But at the same time, there are different neurons that can represent Malleshwaram, Yeshwanthpur, and different parts of the entire map of Bangalore. And just by looking at the activities of these neurons, I can tell you exactly where I am in this environment. So now you have you've heard what the hippocampus does. How does the hippocampus creates a representation of space by firing at different neurons fire at different locations in order to create that map. How does the hippocampus create that map, and what can this map be used for? Let's answer these questions now by looking at what do the inputs to the hippocampus tell the hippocampus. Remember I told you about lateral and medial entorhinal cortex being the two major inputs to the hippocampus. So, and I told you that path integration and movement derived spatial information comes in via the medial entorhinal cortex, and external sensory input derived information comes in via the lateral entorhinal cortex. So I'm going to tell you what these regions do in spatial navigation tasks. So here, for example, are recordings from Moser lab in the medial entorhinal cortex. As the rat forages around, this one single neuron in the medial entorhinal cortex fires at multiple locations. So these gray lines represent the trajectory of the animal, and dark black spots represent action potentials of a neuron. So this neuron fires here, it fires here, it fires here. So unlike the place cell in the hippocampus that fires at one single location, this grid cell in the medial entorhinal cortex fires at multiple locations. But these multiple locations are not randomly distributed in space, but they form repeating patterns of these equilateral triangles or hexagons that tessellate or tile the entire environment. And as you go from more dorsal to ventral part of the medial entorhinal cortex, you see neurons with larger and larger spacing, grid cells with larger spacing. So they are still grid cells, they still form these hexagons, but the sizes of these hexagons are larger. Each individual vertex is also larger as you go from smaller scale to larger scale. But more importantly, the space between the vertices is larger. This is the largest recording from this particular animal in their lab. So you can go from tens of centimeters to of the order of about a meter or so, meter or more in terms of spacing, right? So this path integration function can create a representation of space, but this is more of a graph paper like representation of space, which represents spacing and directions. And the path integration process itself is basically a process of keeping track of how fast you're moving and which direction you're moving in order to create a representation of where you are with respect to where you started from, or some abstract reference point. So this kind of grid cell representation in the medial entorhinal cortex is thought to be a product of path integration process, given the kind of inputs it gets. But the exact process by which you generate these grid cells is still currently under process of intense study. So that's what medial entorhinal cortex does. In this same experimental paradigm, wherein you put in the rat in a plain environment without anything inside it, if you just allow the rat to run around, the lateral entorhinal cortex does not show much of spatial selectivity. The neurons fire very weakly and they fire all over the environment. So the question arises, what does the lateral entorhinal cortex do? Given the inputs to the lateral entorhinal cortex, it was hypothesized that lateral entorhinal cortex represents a representation of sensory inputs that are coming in. External sensory input derived representations are being created in the lateral entorhinal cortex. So to test this hypothesis, I conducted an experiment in which I allowed the rat to forage around for food in similar environments to before. But now I've placed multiple toys or objects inside this environment. Each of these toys, as you can see, has different colors, textures, shapes, softness, taste, odors, and so on. So they can be very easily distinguished from one another. After the rat gets familiarized, I start doing manipulations like introducing a new object or manipulating objects by moving objects from their familiar location to new locations and then going back to the original location and so on. The expectation here is that because lateral entorhinal cortex gets sensory inputs, it should show you correlates of that kind of sensory information as the animal forages around. And sure enough, we get neurons that fire at objects. So for example, this particular neuron fires at all these 4 objects. When you introduce a new object, this neuron fires at the new object. When you move the object to a new location, the neuron fires at the new location of the object. So, but in addition, it starts doing something much more interesting now. So here is another neuron, starts with something similar, fires at multiple objects. But now when you move the object to a new location, not only does this neuron fire at the new location of the object where the object currently is, but it also fires at the location where the object used to be. Now, this is not a simple sensory representation because you remember that in this particular session, there is no object at this location. There is no sensory stimulus at this location available to tell the rat this is where the object is, because the object is here now, not at the original location. And yet this neuron also fires at the original location. So now this neuron is creating a conjunctive representation of space and object. So this is where an object used to be in the past. So this is representing a misplaced cell kind of activity. So as you can see, the lateral entorhinal cortex is not just creating sensory representations, but it is also creating conjunctive representation of space as well as sensory input, or where the object used to be, precise. Surprisingly, in this experiment, completely unexpectedly, we started seeing neurons that look like classic place cells in the hippocampus. Here, for example, is a neuron that fires at this location, and it fires repeatedly at this same location across multiple sessions, even when an object is introduced elsewhere, or when an object is moved pretty much close to the center of the place field of this neuron. This activity is very different from the object-related activity, as you saw, because this activity is happening farther away from objects, and it's not really changing in response to object manipulations. Here is another example. This activity arises only in presence of objects. When there are no objects in the environment, we don't see this kind of activity, both in terms of previous experiments as well as controlled experiments. When this experiment was done, we did not see this kind of spatially selective activity in the absence of objects. So now here is where we are. Just to quickly summarize at this intermediate stage what we've heard so far. Hippocampus creates a representation of space with those place cells that you heard about, neurons that fire at individual locations. It gets inputs from medial entorhinal cortex, which has this grid cell, which is tessellating patterns of equilateral triangles or hexagons covering the entire space, creating a graph paper-like representation, which can be used for you creating a matrix representation of space and doing simple calculations of how far you've moved, which angle you've moved in, and so on. Lateral entorhinal cortex, in contrast, I showed you, has a representation of objects, and it has object-dependent representation of space. So lateral entorhinal cortex has external sensory information derived representation of space. Now the obvious question arises, does the lateral entorhinal cortex simply inherit this representation of space from its major input, the perirhinal cortex? So we recorded from perirhinal cortex, and sure enough, we saw representations of objects, like neurons that fire at objects in the perirhinal cortex, but not this kind of representation of space, even in presence of objects in the perirhinal cortex. So just to connect this to the previous talk, perirhinal cortex gets quite significant information from the equivalent of inferior temporal cortex in the rat. So object-related information pretty directly gets strongly, and it also gets olfactory and somatosensory information, which would be directly relevant here. But still, it cannot create this representation of space in presence of objects. So it looks like representation of space arises for the first time in this pathway in the lateral entorhinal cortex. It might be either generated de novo by using this information derived from the objects, or it might be inherited from lateral connections from the medial entorhinal cortex or feedback connections from the hippocampus, but it is expressed only in the presence of objects. In absence of objects, you just do not see this kind of place cell representation in the lateral entorhinal cortex. When this information, the grid cell information from the medial entorhinal cortex and object-derived non-spatial as well as spatial information from the lateral entorhinal cortex gets to the hippocampus starts now creating more and more complex representations. I'm giving you two examples over here. One is here is a misplaced cell. This is a neuron that fires at these three different locations where the object used to be. So I'm not showing you the entire sequence of experiments here, but this neuron was recorded when the object was placed at this location first. Then the object was moved to a different location, finally to a third location. And finally, in this particular session, the object was completely removed. So there is no sensory information available for the animal to know where the object is. And yet it's using its memory of where the object used to be to create this kind of representation of where the object used to be. And this neuron is representing all the three locations where the object used to be in the three previous sessions. So hippocampus clearly is using this object-derived information from the lateral entorhinal cortex in order to create object location memory representation here. Hippocampus also has this another representation, which is the landmark vector representation. I showed you classic place cells earlier, which fire at one single location in a given environment. But now there are some neurons that fire at multiple locations. And when you look at their activity, you see that these locations are not just randomly distributed in space, but they can be defined in terms of distance and directions from individual objects. And this representation happens more often than you can expect by chance. So therefore now the hippocampus is creating representations in multiple reference frames simultaneously. Here, for example, these two place fields of the same place cell happen to represent space in the coordinates with this object as an origin for this location and this object as an origin for this location. So, till this point, you should get an idea about how hippocampus has spatial map, but this spatial map is not a very simple static representation. As we made the environment more complex by introducing more objects, it started getting more complex inputs in terms of object representations as well as spatial representation derived from objects. And that representation now has started having influence on hippocampal representation itself in terms of memories of where the objects used to be. So now you can see that this can start feeding into that episodic memory kind of stories, wherein single sessions where the objects used to be now have created memory representations in the hippocampus, and therefore you can see individual neurons representing where the objects used to be, as well as object derived spatial representations as seen by these landmark vectors, which can be used again for navigation very easily, right? Turn left at Gandhi Bazaar or whatever. That kind of information can come in via these landmark vector cells in the hippocampus. Okay, so now I'm going to change the gear and talk about making the environment, changing the complexity of the environment a little bit, and how the representation in the hippocampus changes, representation in area CA1 of the hippocampus changes, and we'll discuss how it can possibly arise from its inputs. Okay, so we are going to look at recordings that are done along the entire proximal distal axis of area CA1 of the hippocampus. And you get inputs from medial entorhinal cortex going to the proximal part of area CA1 and inputs from lateral entorhinal cortex predominantly going to distal part of area CA1 of hippocampus. Simultaneously, it receives inputs from area CA3, which are also kind of segregated. Distal part of area CA3, which has strong recurrent connectivity, tends to project preferentially to proximal area CA1, and the proximal part of area CA3, which does not have very strong recurrent connectivity, tends to project to distal part of area CA1. There are also other inputs, but these are the two major inputs to area CA1, and therefore people predicted from this, given the lateral and medial entorhinal cortical segregation, people predicted that distal part of area CA1 of the hippocampus should not have strong representation of space, while proximal part of area CA1, because it gets strong spatial information from medial entorhinal cortex, it should get strong spatial representation. And here are rate maps in proximal, intermediate, and distal CA1. So these are consecutive sessions showing you that representation is stable. So you see that you get a classic place cell in the proximal CA1. In distal CA1, you tend to have multiple place fields and at the same time larger place fields. And that you can see summarized in this bottom row over here. Proximal CA1 preferentially has single place fields. Individual neurons have single place fields, whereas there are fewer and fewer neurons having more and more place fields within this same environment. Whereas distal CA1 tends to have median and mode at 2 place fields per cell. And that distribution has shifted very clearly rightwards. The spatial information content is significantly higher in proximal CA1 than distal CA1. And other measures like coherence as well as correlation across sessions from first trial to the next or a later trial decreases as you go from proximal to distal. So the conclusion there was that LEC does not, lateral entorhinal cortex does not represent space, and hence its target, the distal CA1, has weaker spatial code. But the question is this always true? You already saw that when you make environments more complex, lateral entorhinal cortex also has complex representations, more spatial representation as well as non-spatial representations. So consequently, an obvious question is, do you now start getting representation of space that is stronger in distal part of area CA1 simply by increasing the environmental complexity a little bit? So here is the experiment that I conducted. I allow the rat to run around on this circular track, which has 4 very, very distinct textures. So animals can distinguish these textures visually, by touch, as well as by smell, because these different textures have different absorbent properties, and therefore different smells adhere very differently to these different textures. So it's very easy to distinguish between these textures. But in addition, there are also multiple visual cues, multiple very large visual cues available along the curtain at the periphery, these global cues. So we'll refer to these as distal or global cues here and local cues on the track over here. So right off the bat, so the experimental paradigm is interleaved with these standard sessions where the local cues as well as distal or global cues are in their familiar configuration that the rat has been trained with. You interleave these with manipulation sessions wherein you rotate local cues by a certain angle and global cues by exactly the same angle in opposite directions. So for example, in order to generate this 180-degree mismatch, your local cues get rotated by 90 degrees counterclockwise, and your global cues get rotated by 90 degrees clockwise. Therefore you get 180-degree mismatch. You can create other mismatch angles also by rotating the two sets of cues in opposite directions by equal magnitudes. And then you interleave those with standard sessions, and we'll see how these things differ. But before that, let's just look at what happens in the standard session. Remember that I told you earlier that the spatial information was highest in the proximal area of CA1 and smallest in the distal area of CA1 in a simple environment without any discernible textures or objects in the environment. However, the minute there are textures in the environment, now there is no distinction between proximal CA1 and distal CA1. As you can see, all these patterns are similar. The histograms of mean firing rates, peak firing rates, spatial information scores, number of place fields per cell, sizes of the place fields, fraction of the environments that are occupied by place field, and so on. No matter what parameters I use, these are the histograms look very similar to each other, and these data are not statistically distinguishable from each other. So simply by changing the environment, we've changed the amount of information represented in the hippocampus. In proximal and distal parts of area CA1 of the hippocampus. So this already puts a big question mark on the previous understanding in the field, which was simply that, oh, proximal CA1 receives inputs from medial internal cortex and therefore is spatial, and distal CA1 receives inputs from lateral internal cortex and therefore it's not spatial. Now the understanding has to be more nuanced. We need to say that proximal CA1, because it receives inputs from the medial internal cortex, can create highly selective spatial representations in absence of too many sensory stimuli. But the minute even simple sensory stimuli like textures are provided, the spatial information in distal CA1 becomes comparable to proximal CA1. So it's not like distal CA1 always has lower spatial information than proximal CA1. So now let's start looking at how these different areas respond to cue manipulations. Here are some examples of neurons. Color code is different, but it's the same thing. Here is a neuron that fires at this particular location on the track, whereas most of the track it does not fire. Here is that same neuron after the cue rotation. And as you can see, this neuron rotates clockwise when the cues are rotated, and therefore it's rotating together with the global cues which have been rotated clockwise. And in the consecutive standard session, it again comes back to the original location where it used to fire. In contrast, here is another example of a neuron that used to fire at this location, but rotates counterclockwise with local cues, and then in the subsequent session, it comes back to original location where it used to fire. So neurons can go with global cues, neurons can go with local cues, and then you can have neurons that do something ambiguous. So here, for example, this neuron starts with firing here, but now in presence of cue conflict where global cues have rotated clockwise, one field is formed with corresponding to that location, global cue rotation, and another field is formed with corresponding to the local cue rotation, and therefore you get a split representation in the same single neuron. So this representation is rather ambiguous. And then you can have other representations wherein a neuron that does not fire on the track now suddenly starts firing at a particular location, or a neuron that has a nice place field on the track goes to not having any place field or not firing at all on the track. And then subsequent session, it reverts back to whatever the activity was in the standard session. So you get all kinds of these responses. So now we can ask, how does the ensemble react together? So in order to do that, we need to look at 2D correlation matrices. So I'm going to take a minute here to explain the methods here so that you understand what I'm showing you in the 2D correlation matrix. Each of these rows over here corresponds to activity of a single neuron as the rat traverses through 0 to 359 degrees. So the entire circle on that circular track is a row over here. Here is neuron 2 row, neuron 3 row, neuron 4, and so on. So if I look at each column over here, that's the activity of all the simultaneously recorded neurons 1 through n at a location 0, the bin 0. Here is activity, population vector activity at bin 1, bin 180 degrees, 359 degrees, and so on. So now you can take these bins and find out correlations. How strongly does the 0 bin of standard session 1 correlate with the 0 bin of standard session 2? If the representation is stable across these two standard sessions, the 0 bin should show strongest correlation with the 0 bin. One degree bin should show strongest correlation with 1 degree, 180 degrees should show strongest correlation with 180 degrees, 359 degrees should show strongest correlations with 359 degrees, and so on. And therefore, this correlation matrix that is created should show a band of highest correlation at 0 degrees. So now instead of having standard 1 versus standard 2, if you were to create correlations between standard 1 and mismatch session, if the representation coherently rotated with global cues, you should see a band of high correlation that is displaced from this diagonal in the direction of global cues. If the representation moves with local cues coherently, you should see a coherent band that is displaced from the diagonal in the direction of the local cues. If, on the other hand, the representation is no longer coherent, you should see activity that is firing all over the place. And so on. So we'll see what happens in different parts. Medial entorhinal cortex, when you look at standard versus standard, you see diagonal representations. The highest correlations are right around the diagonal. But when you look at standard versus mismatch at different mismatch angles over here, what you see is that the band of highest correlation gets farther and farther displaced with increasing mismatch angle from the diagonal, indicating that the representation rotates coherently with the global cues. Lateral entorhinal cortex, as I told you, in simpler environments doesn't show very strong spatial representation, and correspondingly, even the standard versus standard correlations don't show a strong band at 0 degrees. However, when you look at standard versus mismatch, there is a slight preference for rotating with the local cues. It's very minor, but it's statistically significant, so we have to pay attention to that. In contrast, when we look at the other input, you remember we are interested in what's happening along the proximal distal axis of CA1, but we are looking at what the inputs are doing. I just showed you medial entorhinal cortex rotates with global cues, lateral entorhinal cortex weakly, but statistically significantly rotates with local cues. In contrast, now when we look at what the area CA3 is doing, in standard sessions, distal CA3 again shows very strong band of correlation along the 45-degree angle. But in the mismatch session, the band of high correlation tends to move with the local cues. And it's a fairly strong band of high correlation that moves with the local cues with different mismatch angles. Whereas proximal CA1 tends to pretty much just fall apart. You see hotspots of correlation, sometimes with local cues, sometimes with global cues, and so on. There is no single, simple, coherent kind of representation that you see over there. Now let's see what happens in area CA1 of the hippocampus. Proximal CA1, remember, it receives two very strong conflicting inputs, right? Medial entorhinal cortex is telling it to rotate with global cues, and distal CA3 is telling it to rotate with local cues. So there is global versus local kind of conflict going on. And sure enough, that conflict is reflected in the activity of proximal CA1. You see split representation. There are hotspots along global cue rotation as well as hotspots along local cue rotation in proximal CA1. At different mismatch angles, you see the same thing. As against that, if you look at distal CA1, there is a very clear strong band of rotation along global cues, but weak hotspots along local cues. Here, strong band along global cues and weak hotspots along local cues, and so on. So now here we are, right? Activity of proximal CA1 can be explained as, okay, sure, you get conflicting inputs and therefore the activity gets split. However, remember that both lateral entorhinal cortex as well as proximal CA3 are giving very weak locally rotating signal to distal CA1. And yet distal CA1 tends to fairly coherently rotate, but coherently rotate with global cues, not with local cues. What the hell is happening here? How did we get at this point, right? So then you have to consider that there are other inputs that are generally weaker in the kind of spatial configuration that we are seeing. But when the main inputs of lateral entorhinal cortex and proximal CA3 are not giving a very strongly coherent information, other weaker inputs might be sufficient to drive a very coherent activity in distal CA1. And the input to consider over here is nucleus reunions, which sends which has head direction related information in there. And we know that the head direction system very coherently rotates with distal or global cues over here. So this is a different representation because this came from a different paper from the same lab. Herein, they're just showing you activities of individual neurons. So if they rotate with, if standard versus standard, it remains at 0 degrees. So most of the neurons show you strongest correlation at 0 degrees. But when you look at standard versus mismatch correlations, those tend to rotate with distal or global cues very coherently, and very few neurons tend to rotate with proximal cues, local cues. And you have that kind of information coming from nucleus reunions to area CA1, distal part of area CA1. So that might be sufficient to drive distal CA1 to very coherently represent space. So now remember, we started this proximal distal CA1 story with saying that that proximal CA1 was thought to be more spatial because it receives inputs from the hippocampus, from medial entorhinal cortex, and distal CA1 receives from lateral entorhinal cortex, which is weakly spatially selective, and therefore we didn't expect much spatial selectivity here. But now we've ended up at a point where when there is cue conflict, distal CA1 is the one that is maintaining a coherent spatial representation, right? And proximal CA1 is pretty much falling apart. So you have to keep in mind that the representation of space in hippocampus itself is dependent on task demands as well as degradation of the environmental inputs that are available. Some parts are easier to confuse, like proximal CA1, than the other part, which is the distal CA1. I'm going to quickly skip over this. This part is basically just to tell you that Corresponding to spatial information, you also saw difference in oscillatory activity in proximal and distal CA1. The distal CA1 showed lesser spatial selectivity than and correspondingly lesser theta modulatory activity in the open field tasks before. But in our hands, when we have these texture tracks, we actually see that distal CA1 local field potentials actually show stronger theta power than proximal CA1, and distal CA1 individual neurons show as much modulation by theta as proximal CA1. So, and then theta phase precession, I won't get into that, but it's timing. Sorry, so my 50 minutes are up, so I'm just going to wind this up here quickly. So you see very coherent kind of phase precision happening in proximal as well as distal CA1, which allows neurons to have a temporal code. So quickly summarizing this part, distal CA1 can be more spatial than thought earlier. Distal CA1 shows coherent rotation with global cues, while proximal CA1 becomes disorganized. You saw that proximal CA1 can act as a mismatch detector. The representation splits when expected spatial location coming in from CA3 disagrees with current spatial location coming from medial entorhinal cortex, and therefore CA1 is capable of acting as mismatch detector. And finally, distal CA1 shows stronger theta oscillations and stronger theta modulation and phase precession there. And then just to summarize the overall picture that I told you, hippocampal place cells create a spatial map. Complexity of the environment affects the kind of representation that is seen in the hippocampal inputs, lateral and medial entorhinal cortex, as well as the hippocampus. And you get a rich representation of space in activities of place cells in the hippocampus, grid cells in the medial entorhinal cortex, object cells, mismatch cells, and landmark vector cells in lateral entorhinal cortex and the hippocampus. And this indicates that there are multiple maps of space with varying details coexist in the hippocampal system, and they can be activated depending on the demands of the tasks. And then finally, spatial information processing along the proximal distal axis of CA1 changes depending on task demands and may rely on different inputs depending on the task. All right, so I'll just keep this acknowledgement slide over here. I've talked about some work from different people in the lab. Mekhla's work I was planning to present, but I didn't quite get around to it. Mekhla's and Indraja's work. So if there are questions, I can happily take this now.